Okay, we are on page 244. Turn to 244.
I've been at 244 for the past 35 minutes.
Whoa, is that coming in?
And we're talking about cell reproduction, this chapter, and cell size. Now, our first question is, why do you have so many cells? Why not just have, we have 80 trillion cells, why not just have 1,000 cells and have each cell real big?
Can't do that.
There's a problem with that.
Jesus won't let you.
The major problem is something that we will call surface area to volume ratio.
Yeah.
Surface area to volume ratio. Now, let's talk about what surface area means. What does that mean? Here I have a dice. A die. Where's the surface? What is the surface of the die?
What?
What is that? What am I talking about when I say it's the surface?
The numbers.
I'm talking about what I can touch, right?
Yeah.
What's on the sides?
You look nice off.
You could calculate the surface area if you knew how long each side was pretty easily.
You roll dice.
The comments again, Taylor. You're making comments that aren't necessary for my lecture.
Yes, sir.
Hey, bring it up.
There's more. That's more comments from everybody. So this is the surface. What is the volume of the die?
How much is inside?
The volume is the stuff inside. So the die has two aspects: the surface area and the volume, what's inside. And if you were to mathematically calculate the surface area and divide it by the volume, you would have the surface area to volume ratio. Now, what cells need is a surface area to volume ratio that is very high. And what that means is the surface area needs to be high and the volume needs to be low. How is that possible to get a lot of surface area and very little volume?
A lot of cells.
Well, the easiest way to get a lot of surface area compared to your amount of volume is to make yourself smaller. This cell right here is real big. It's got a lot of surface. I mean, it's got a good amount of surface, but it's got a whole lot of volume. This cell right here has very little volume, and it still has a good amount of surface. I think it's easier to show with the book with the picture the AP biology book uses. Let's take a look at this. Here's one cube. Imagine its surface. You could touch its surface, right? This is the same cube cut into 64 smaller cubes. So this on the left and this on the right have the same volume, don't they?
Yeah.
The amount of stuff on the inside is the same for this and this. But which has more surface?
The one on the left.
Little one?
64 little cubes have a lot more surface than one big cube. Don't they? If I cut this up into 64 smaller cubes, think of all the extra surface there would be. It'd be the same amount of volume, but a lot more surface. Cells need a lot of surface. Why do cells need a lot of surface? Because surface is where you take in your nutrients and you get rid of your wastes. The more surface, the better. So a cell that's real big just doesn't have enough surface to get rid of all of its waste. But if I were to cut the cell into real small cells, then there'd be lots more surface for the same amount of volume. You see? These cells, let's say that was an individual cell. It has to get rid of its waste and take in food, and it does that through its surface. And imagine there's cells on the very inside too. You're giving them extra surface if you make the cell small than if it's one big cell. So cells need a high surface area to volume ratio. And that's why cells are small. The smaller you get, the more surface area you have compared to your volume. Okay? Also, the process of diffusion is very, very slow.
Your computer's
The process of diffusion is very, very slow. Molecules take a long time to move from the outside to the inside if the cell is this big. It might take, if a cell was this big, it might take days for molecules to go from the outside to the inside.
That's a lot of ATP.
So, if you make the cell smaller, if stuff hits the surface and then travels real slow before it finally gets to the middle, if you make the cell small, it's to the middle a lot faster, traveling at the same speed. So, the book talks about that, transport of substances, on page 245. The smaller the cell, the faster stuff can get in and out of it. Because you don't have as far to go to reach your surface. Do you see? And one other reason cells have to be small, the nucleus can only control a certain amount of cytoplasm. A certain amount of stuff going on. You know, the stuff in the cell, what goes on in the cell is controlled by the nucleus. So the nucleus can only control stuff that's kind of close to the nucleus. If you were an organelle, like a Golgi apparatus way out here, it would be hard for the nucleus to get instructions to you. Or maybe a ribosome way out here. It'd be hard for the nucleus to get instructions to you. Because the instructions would have to travel all this distance. Make the cell small, nucleus can control everything in the cell very easily. You see? Everything's close to the nucleus in this cell. A lot of stuff is not close to the nucleus in this cell. Yes?
I like to go to class.
Okay.
Okay.
So, the result of all these reasons is when the cell gets pretty big, it then has to divide and make more surface for itself. And that's what cells do. Once they reach a certain size, they divide. And they go through what's called the cell cycle. And the cell cycle consists of basically 5 phases that you have to memorize. G1, S, G2, M,
C.
and C.
This cut.
G1, S, G2, M, and C. All right?
Are we going to explain what each one means?
Absolutely. G1 is the growth phase of the cell. That's when a cell is getting bigger. It's starting to grow. Cells start small, just like anything, and get bigger and bigger until they reach a certain size. Then they divide. So G1 is the first growth phase. In a quickly dividing human cell, this G1 phase lasts about 8 hours. And that's when the cell is growing and getting bigger. That's why it's called G. G stands for growth. Cell grows and performs normal functions, whatever a cell normally does. If a cell is in your mouth and its job is to make mucus, that's what it does during G1. It makes mucus. Once the cell gets big enough, it decides it needs to divide. It goes through what we call the S phase, or synthesis phase. The S phase is when the cell copies its DNA. There's DNA. Where's the DNA in the cell?
Nucleus.
In the nucleus. And that has to be copied. Why would a cell have to copy its DNA?
To make use
It
of
has
it.
more information.
Because it's going to divide in a minute, and each cell needs a copy of the DNA. The S, the synthesis phase, takes about 6 or 7 hours in a quickly divided human cell. So now we've copied our DNA. Then there's a G2 phase. They called it G because G stands for gap, because it seemed like just a length of time where nothing was really happening in the cell, where the cell was just kind of sitting there. Now they know that there's some more growth going on inside. The cell is actually producing proteins that will be used, and enzymes that will be used when the cell divides. So the cell is preparing for mitosis. Mitosis is the division of the nucleus of the cell. That's M. Mitosis. And we're going to spend all day tomorrow talking about M.
Sweet.
Mitosis itself has four phases. Prophase, metaphase, anaphase, telophase. Did y'all learn about this in life science?
We sure did.
No.
We have. Jay, what was cyto?
Could be like anything.
Now, cytokinesis, mitosis is the division of the nucleus. That's where the nucleus of the cell breaks in half. That's mitosis. Cytokinesis is the division of the cell. Because after the nucleus breaks in half and you have two nuclei, then the cell will divide in half.
Yep.
And you'll have two cells. So that cytokinesis is when the cell divides in half. And we call this whole set of activity that goes on the cell cycle. Because after cytokinesis, you have two new cells, and then each cell is back to the growth phase. So once you divide, each cell gets bigger and bigger, and then each cell will divide again. And you start with one cell, and you have two cells. At one time, you were one cell big. Then you divided, and you had two cells. Then 4. Then 8, 16, 32, 64, 128, 256, 512, 1024, 2048, 4096, 8192, 16,384. 32,768, 65,536, 131,072.
Do you like time yourself on this for fun?
No.
Bro, what's the difference between synthesis and
Absolutely no way to
mitosis?
do that.
Good question. Synthesis is where just the DNA is being copied. In the nucleus, you have a bunch of DNA laying around, and you got to copy that DNA.
Okay, so it's getting ready.
So it's still one nucleus, but now after the S phase, there are two sets of DNA in the nucleus.
And mitosis is supposed to
And then mitosis is when the nucleus itself splits, and this now there's one section of DNA over here and one section of DNA over here.
Gotcha.
Do it, Hogan. Good questions.
God, it's so hard to move.
Tucker!
You unscrew that.
Now,
Screw people's names.
Screw that thing a little bit and it makes it work. He's got his mouth. Okay.
I want to introduce you. I got to go to the AP section for this. I want to introduce you to a structure.
Can we look at your Willis family photos?
No.
Yeah, maybe one. Maybe.
Do you have like nieces and stuff? Or nephews? Or?
I got. No, I don't have any nieces or nephews.
Any kids?
He's
got a father and a mother.
He's got 3 kids. Crazy question. No, he doesn't.
Does he?
Yeah.
What? No, that was right.
Why so serious?
Nothing for you.
Here we go. Video footage of the cell cycle
Yes.
Yeah, man.
No.
How fast does that happen?
takes about 24 hours for the whole thing.
Is
The
that what
process
is
of cell
replaced?
growth and division
hold on.
in eukaryotes is called the cell cycle. This cycle is divided into phases based on what is happening in the cell at a given time. A cell grows during the G1 phase. During this phase, there is a chemical checkpoint that controls whether the cell will divide, delay division, or enter a resting stage. When conditions in the cell are right, the G1 checkpoint will be passed and the cell will enter the synthesis, S phase. During the S phase, DNA replication occurs so that future cells will each have a complete set of the genetic instructions in the DNA. After DNA replication is complete, cells enter the G2 phase, where they continue to grow and prepare for cell division. At a checkpoint in this phase, the success of DNA replication is assessed. If all is well, the cell enters the mitosis, M phase. During the M phase, a complex series of events moves the DNA so that a complete set of genetic instructions will be sent to each daughter cell. The process of mitosis is assessed at a checkpoint during the M phase. Once this checkpoint is passed, the cell will complete mitosis as well as begin the cytokinesis, C phase. Part or all of the C phase actually overlaps with the later parts of mitosis, so it is not a distinctly separate phase. During the C phase, the cytoplasm of the cell is divided, and two daughter cells are created from the original cell. When this process is finished, the daughter cells enter the G1 phase, and the cycle is complete.
Question?
Yes.
What happens if the G2 checkpoint, everything's not fine?
Ah, good question.
You see what I'm saying?
Yes. If at the G2, shh. She asked me a question. Now, why are you guys answering?
Because it was a dumb question.
Why are y'all talking then? It seems to be the same couple of people
I was answering a question.
that are talking.
You're not looking at me.
Like right now. Seems to be that. Just stop.
Look at me.
At the G2 checkpoint or the M checkpoint, and sometimes at the G1, if everything is not right with the cell, a cell will kill itself. It's called cell suicide. It's known as apoptosis. And cells do this. And that's important because if something's wrong with the cell, you don't want that cell to continue growing because remember, a single cell will divide and copy itself exactly. And so if something's wrong with one of your cells while you're growing, that messed up cell could become millions of messed up cells if it continues to divide. So cells have a way of combating that. Whenever they reach a spot in the checkpoint, something's wrong with them, they kill themselves. Now hold on. There are some times where the checkpoints screw up and the cell doesn't kill itself. And sometimes the cell is supposed to stop, like at the G1 checkpoint, it's supposed to stop there, but it doesn't. It just runs through its checkpoints. And it continues to divide over and over and over again without wanting to stop dividing. We call that cancer. A cancer is a cell that is going through the cell cycle without stopping. And it becomes a big lump of cells that we call a tumor. And not tomorrow, but the next day, we're going to talk about cancer because that's what it is. Cancer is a screw up where a cell does not stop reproducing, where it just continues to do so. Okay, yes?
So, what happens when the cell kills itself? What happens to the dead cell?
It's eaten up by white blood cells. You have cells in your body that go around and clean up any dead cell parts. White blood cells.
Oh, gotcha.
Yes.
Okay, so say you had like a wild night one night.
Okay.
And you woke up.
Yeah.
And you had to go get one of those little things where if you're gonna have a baby, it like kills it. Does it like kill the cell?
An abortion?
No, the thing that's called like a Plan B thing.
Yeah, it
That's
does.
on TV.
Yes, it kills the cells.
But like, or like you just like kill them.
Yes, I think so. I think it's got some chemical in it that kills the cells. Yes.
Well, okay. So, when you have cancer, the
Yeah.
white blood cells, you have a fewer amount of white blood cells.
If you have cancer of white blood cells, that's called leukemia.
No, but when you have cancer, like all types of cancer, don't you have less white blood cells?
No, not necessarily.
Just leukemia?
Leukemia, you have more white blood cells. It's the white blood cells that are reproducing out of control.
Oh.
But one thing about leukemia is when they're reproducing out of control, they don't do their jobs. And so you have thousands of, or way too many white blood cells, and they're not doing their job. That's a bad problem. Yes?
You said whenever it doesn't go through checkpoints, it's like probably wrong. And it makes bad cells. Is that how we get like disorders or the way we like,
Sometimes, yeah, you can get screwed up cells that something's wrong with and they haven't killed themselves. Yeah, that could be a disease. Yes.
In the video, that said daughter cells. Is that like the cells that just were like split?
Yeah, after it splits, we call the two daughter cells.
That is, yeah.
Good question.
Doing that.
Okay,
Earthquakes.
Oh my
here's what it looks like when a cell kills itself. This cell, something's wrong with it. And it becomes round, and the nucleus disintegrates, and the cell comes apart into pieces.
And then the white blood cells clean it up.
And the white blood cells clean up the dead pieces. That's called apoptosis.
What are the
Cell suicide.
Bro, what are the other 4 cells in the very beginning? Why are they so
These are normal cells that are behaving normally, and this one's messed up.
Oh, it's just showing them hanging out.
Why
Yeah,
is it
it's
messed
showing
up?
hanging.
How can you tell it's messed up?
They're just telling me that it
Look at it.
You can't tell. Something's wrong with it.
Something is wrong with
There's a
it.
bomb inside it.
Okay, this is what I wanted to show you. Listen, let's discuss the DNA. In a nucleus, let's pretend this room were the nucleus of a cell.
Okay.
In the nucleus of a cell, there's DNA laying all over the place.
Like all of us.
Sort of.
Kind of.
But DNA is more like a long string.
Oh.
Remember, it's nucleotides attached together in a chain?
Gotcha.
Do y'all remember when we studied DNA?
I do.
The A's and T's and C's and G's, and they form like a double helix, like a ladder. Like that on top. Now the DNA is actually laying in the nucleus. Taylor, pay attention here. The DNA is laying in a nucleus in a long string. It's like a piece of fishing line. Except that it's not just one piece of fishing line. Imagine a long fishing line laying in the room. There are actually, in humans, 46 pieces of fishing line laying in the nucleus. But it's not fishing line, it's DNA.
What?
So there are 46 strands of DNA laying in the nucleus, and they're real long. Imagine a whole, like if you're a fisherman, you know how much fishing line there is on one spool. Imagine if you pulled off all the fishing line on one spool and laid it in this room, and then you did that with 46 other fishing line.
Wow.
So you'd have, but it doesn't lay on the floor, it kind of floats in the nucleus, so it's all in the room, fishing line everywhere. Wouldn't it be a mess?
Yeah.
Imagine having to copy each piece of fishing line exactly, and then move half those copies to one side of the room and half the copies to the other side of the room. That's the task we have if we're going to do cell division. And the way, what would you do if you had a long piece of fishing line that was laying in the room and you had to move it somewhere? What would you do?
The centriole doesn't pick it up and just put it somewhere.
Would you just pick up the end of it and walk over here like this?
Of course not.
What would you do?
Fold it up.
You'd spool it. Wouldn't you? You'd spool it up.
Protein.
You'd take you'd attach it to your fishing pole and do this until it's a small batch, and then you can move that pretty easily, couldn't you? Hold on just a second. This is showing how a chromosome is what the fishing line is called. When the fishing line wraps up, when the DNA wraps up and coils up, it becomes what is called a chromosome. A chromosome is a bunch of DNA all coiled up, so it can be moved. And this is showing you how the DNA, this is the DNA here, okay? And what that DNA, we're panning our camera back. See, that's this DNA right here, and it's in a long line, and it's actually wrapped around proteins in the nucleus. And these proteins will come together to coil up the DNA, eventually into a structure that we call a chromosome. This is a chromosome, a bunch of coiled up DNA and proteins. And do you see that there's two copies? This and this are exact copies of one another.
Is that in the nucleus?
Because the DNA was copied during S phase. There are 46 of these in the nucleus when the cell is about to divide.
Okay.
46 strands, and they're exact copies.
So there's 7.
So there are 47.
Things.
No,
92.
there's
There would be
48.
92. We call that a chromatid. and that a chromatid, and since they're exactly alike, we call them sister chromatids.
Wait,
there are 92 sister chromatids, 46 chromosomes.
Why not brother?
I don't know, they just call them sister. This is a great video I have showing this DNA coiling up.
Is it real?
Now watch it. Now this is computer animation. This line right here, this is the DNA. And the DNA is actually wrapped around proteins. There's a protein right there. And the DNA wraps around each protein twice. So watch this long strand of DNA coiling up. This is like taking your fishing line and coiling up. This happens right before the cell divides. See how the DNA coils up? Now watch it. And then those loops coil further. They're panning back here. This is one of the 46 strings of DNA that are in the nucleus.
Whoa.
Wait, watch it. We coil it up and wrap it up into one bundle. And that's a chromosome.
What?
And there are 46 chromosomes in each cell, in every cell. Let's watch that one more time because the scientists have figured out how this works, even though these molecules are almost too small to see.
We need some good music.
Watch it coiling up.
Bam.
See how it's very nice and orderly. This coiling DNA.
So is this like one spool of
This is one spool of line. This happens in 46 different lines in the nucleus.
And it's copied, right?
This is right before the cell divides. It does this. So right before a cell divides, you know a cell's about to divide when you look at its nucleus and you can see these dark structures called chromosomes. That means the DNA is coiled up and is ready to be moved.
That's so cool.
And if you look, what we're going to study tomorrow, this is what a chromosome actually looks like under the
Looks
microscope.
like hamburgers.
Are we doing this in the lab tomorrow?
Looks like what I eat at lunch.
We will look at these in the lab, yeah. Not tomorrow.
They're red.
Look at this cell. Listen up. This is what the cell looks like when it's going to start dividing. You see these black things? Those are the chromosomes. The wound up, coiled up DNA. And this is what it looks like under our microscopes. See the little black dots in the nucleus? Those are the chromosomes. 46 of them laying around. This is what it looks like in a plant cell. Look at the chromosomes.
It's like worms.
They look like worms.
You have worms.
That's an onion cell, and that's a whitefish cell.
A whitefish?
A whitefish is a fish.
White.
A baby fish. And we're going to look at both whitefish cells going through mitosis and onion cells going through mitosis. Under the microscopes in there.
Caught a whitefish.
You caught one?
Yeah, in Idaho when I was traveling.
Did y'all eat it? Why not?
Something crap fish.
They're not good?
We caught him in Idaho.
Okay, you can turn it off, Bennett.
Say Willis.
Willis out.